Hello, fellow foodies, and happy October. This week, my book called The Plant Hunter is finally available and can be found on bookstore shelves across the US, Canada, and the UK. In it, I share my life's journey in science, developing new ways to fight illness and disease through the healing powers of plants. It's a book about adventure, scientific medicine, and so much more. To learn more about the book or order a copy, head to my website at CassandraCrave.com. Now, today we have a really great show planned for you. We've been dancing around the ideas concerning the gut microbiome and how microbes that live inside of our bodies are important to health. But we haven't really taken that deep dive yet into the real science of the gut microbiome. And this is why I'm so excited to welcome a friend and collaborator, Dr. Rheinallt Jones, to the show. Dr. Jones is an associate professor at the Division of Gastroenterology, Hepatology and Nutrition of the Department of Pediatrics at Emory University, and his office is actually just a few doors down from mine, so we're office neighbors. His research focus is in identifying the functional elements and molecular mechanisms that mediate the interactions between host cells, especially in the gut, and beneficial microbes, also known as probiotics. His current focus is identifying how microbiome-generated bioactive metabolites that permeate from the gut to every organ and system in the body impact the biology and pathology of the host. Reynold is also the founder and director of the Emory Gnotobiotic Core, where he uses germ-free animals in his studies into host cell and microbe interactions. In particular, he uses germ-free mice to establish the causality of microbiome-associated phenotypes, where he colonizes germ-free mice with a human microbiome and determines if the phenotype observed in the human is transferred to the mouse via the microbiome. Lots of really big terms and lots of really cool ideas, and we're gonna dig deeper in and kind of flesh these out. So welcome to the show, Rheinallt. It's great to see you.
You too, Cassie. Thanks for having me.
Yeah. Well, why don't we start with the basics? Can you define for us what is a microbiome and what types of organisms do we typically find in the gut microbiome?
Right, exactly. So, what I tell people is that every exposed surface of your body is colonized by bacteria, by microbes. I mean, it's really the collection of microbes that are on that exposed surface that is called the microbiome. So, for example, on your skin surface, you have a collection of bacteria, and those would be the skin microbiome. And of course, the part of the body that's most studied with pertaining to the microbiome is the gut. So inside your gut lumen, you have a community of perhaps between 500 and 1,000 different species of bacteria.
Wow.
And together they form the microbiome. So that is basically this term microbiome. It's really the collection of bacteria that you have on any particular exposed surface of your body. And I'm going to add to that in that, remember when I tell my students, the inside of your gut in the body is really outside of your body. So when you think about your alimentary canal, your GI tract, and what's in the middle of your GI tract is really outside of your body. So your microbiome, that's inside your gut, but it's still inside like a pipe inside your body. So that would also be, considered to be outside of the body. So that's the microbiome.
That's fascinating. It sounds really complicated, though. I know microbiologists tend to work on one species at a time, and you're saying that there are huge numbers of different microbes in the gut. So how do scientists go about studying that great diversity in the gut?
So absolutely great question. At about 2005, 2010, there was this leap in our capacity to do DNA sequencing. And so that was really what ignited this microbiome research revolution that we've seen over the past 20 years or so, 15, 20 years. So the capacity to do large-scale sequencing, DNA sequencing of every microbe in the gut or on any exposed surface of the body. And you do some clever DNA sequencing and then run it through a computer program, and then you can ascribe a taxonomical sort of clade to each of the sequence DNA that you sequenced, and therefore you can know that it's a different species or a different type of bacteria that's present in your gut. So you get a measure of how many different bacteria that are there, and the general complexity, the community structure of the bacteria that live inside your gut.
That's great. Well, when we think about the human lifespan, does the composition of your microbiome stay the same from childhood through adulthood, or does it change during your lifetime? And what kind of leads to those changes, if there are some?
Of course, this is a contentious issue, because to determine this properly, you would have to do a longitudinal study throughout an organism's life, right? So, and in humans, we haven't spanned all those years yet. So to do that experiment properly, you would have to take an individual at birth and then examine the microbiome at birth to weaning, perhaps, 18 months old or something like that, as they get the dietary changes. And then you follow the microbiome through adolescence, puberty, then all the way to old age. And you would have to go all the way postmenopausal influences in women. So all these punctate points throughout your life, those could be mark points where the microbiome could change. However, I would say that there is a sort of a consensus, an early consensus, that, of course, you are born sterile. You're sterile within the wound. You're born, and then your first inoculum when you're born comes from the birth canal of the mother. And then you're on a milk diet for the first, say, 12 months, 18 months of life. So you're going to have a bifidobacteria, lactobacillus sort of dominated community for the first couple of years. And then while your food source changes, between the age of 2 and 5, you will establish a microbiome. And there are some schools of thought that microbiome is pretty constant throughout your life.
Fascinating. Yeah. Yeah. Well, when we think about what we're eating, really, we're not just feeding and nourishing our bodies, we're also feeding the creatures that live inside of us, right? Is it?
Precisely, yes. So that early microbiome that you establish with a lactobacillus bifidobacteria, that established because the food source is milk, right? It's the mother's milk. So those bacteria are able to utilize the mother's milk, and they thrive during that period. But as your diet changes, then the composition of your microbiome would change. And just like you say, I tell my students as well, when they're eating, remember that you're not only you're feeding yourself, but what you're actually doing is feeding the microbiome that's within your gut. So every time you take a bite of food from now on, think about what only it does for your body, but what it does for the microbiome composition within your body. And I should say each of those microbes, they have to live, and they get a carbon source, a substrate, an energy source, and then they release little molecules, some of them could be bioactive. So the microbes in your body get their energy sources from the food that you eat. And really, evolutionary, it asks an interesting question. What are you really? Are you really a vessel to carry these gut microbes, these 1,000 different gut microbes around, and go from place to place to forage for food And you're like an analogy that I heard somebody once give was, it's like a bus full of people, and the people would be inside the bus would be your microbiome, and you go from drive-through to drive-through to drive-through, and you're you're feeding the people the microbiome inside you, if you like. So that's one way to think about things.
Yeah. Well, sometimes I know that there can be instances where the microbiome is completely disrupted. And in a one classic example of that can be when someone needs to take antibiotics. So what happens to the gut microbiome when you have the introduction of antibiotics?
Well, just to go back to what I said earlier, is that it's generally thought that the microbiome would be pretty constant throughout your life. But to disrupt that community structure, you need something that's impactful. And broad spectrum antibiotic is one of those things that could disrupt the diversity of your microbiome. So, and of course, you have different antibiotics with different modes of action, right? Some antibiotics target gram-negative, gram-positive, different things. So the impact of the antibiotic on the microbiome depends on the antibiotic that you take. But the best example of this is C. difficile sort of infection.
C. diff infections, yeah.
So that is a terrible, debilitating IBD type disease, right? So what happens normally in that case is that your gut, somebody's been on a broad spectrum antibiotic and has cleared out the microbes from their gut. And then after they come off the antibiotics, it's a race to recolonize the gut. And this Clostridium difficile, this C. difficile, C. diff bacteria is very good at recolonizing and causes this debilitating bacteria. So that would be one sort of effect of antibiotics, because you disrupt the microbiome composition. And just to go further from this, why don't you get C. difficile infection before, right? In normal circumstances? Well, that's a concept called colonization resistance. So once you get this colonization, perhaps between the ages of 2 and 5 of the microbiome, then that microbiome then serves as one of your frontline defenses against pathogens. Those microbes colonize your gut, they stick their elbows out and help defend against pathogens in your body. And, those bacteria that colonize your gut from early on, they've got a great deal, right? They've got temperatures of 37 degrees inside your gut, and then you carry them around and feed them often, right? So they want to make sure that you're healthy, right? That's a part of the symbiotic relationship. So if any pathogen comes along, like, the bad E. coli, the bad salmonella and things like that, then one of your first line of defenses is the microbiome composition of your gut, that they secrete these little molecules that kill the pathogenic bacteria. So it's like chemical warfare inside your gut, in between your symbiotic gut microbes and any pathogen that comes along. So just to go further, just to come full circle on that, when you take antibiotics, you get rid of your natural microbiome, your defenders that elicit this colonization resistance, then you leave yourself susceptible to a more pathogenic sort of infection thereafter when you come off the antibiotics.
Yeah, this brings up a really interesting facet, I think, of the microbiome, because I think we're all, in today's age, we're so fixated on this idea of sterilizing our hands and killing germs at all costs. But actually, as you're describing it, commensal microbes that live in a symbiotic relationship with humans are really important to ensuring our health. And I know from your research approach, you're able to take a really unique look at this because you've actually studied what happens, what types of disease phenotypes can result in the absence of a microbiome. So maybe we can talk a little bit about that, like this idea of what happens if there is no microbiome present and with the immune system and everything else going on with the host.
Absolutely, yes. So I like to frame this story by going back in history and going way back in history. So the universe, what is it, 13.7 billion years old or something like that, right? That's, I think, let's say 14 billion. Okay, so the Earth then is 4 billion years old. And then microbes perhaps appeared on Earth, say 3.5 billion years ago, right? So we have that timeline. Multicellular organisms first appeared perhaps 1 billion years ago. So you have 2.5 billion years of evolution that the world only included microbes, right?
Mm-hmm.
And then 1 billion years ago, you had multicellular organisms appear or start to evolve. So by definition, necessarily, all these multicellular organisms, starting off with little worms and starfish and things like that, they had to have evolved with microbes in the background, right? So it's always been a symbiotic relationship between microbes in the environment and organisms. And as I said, those microbes, they're very clever. They evolve very fast. And they evolved with us such that we give them benefits and they give us benefits. That's the essence of a symbiotic relationship. So bringing it forward to modern day living, we evolved on the plains of Africa for the last 4 million years. So we have evolved a microbiome and has profound effects on our health and disease. Now, the way to examine what is the actual effect of the microbiome on our health and disease is by making an animal, like a mouse, to germ-free status. So basically, it's raising a mouse and completely eliminating the microbes from that mouse. Every exposed surface has no microbes on it. And this technology has existed since about 60 years. So it started in the 1950s. So basically, you can do it because in utero, that's a sterile environment. So if you give a C-section to a mouse and keep the pup inside the uterus, and then if you pass this uterus bead in the mouse through an U-shaped tube, such to sterilize the outside and pass it into a sterile environment, then you open up the uterus and there you have a sterile pup from inside the uterus in a sterile environment. And then you have to rear that pup with germ-free food and everything like that. And then you have a germ-free mouse. So it's sort of, nowadays we use IVF technology into germ-free mice to generate germ-free mice. So technology has advanced considerably to generate these germ-free mice. But the point being is that these germ-free mice, if you think about it, what I just said earlier, it's a situation that's never existed before in a billion years, right? There's never been a multicellular organism that has existed in a germ-free status. So this germ-free mouse gives us an exquisite reductionist sort of model that we can at last find what is the actual influence of our commensal microbes on our gut. And the tactics or the approach that we take is, like I said, if there's between 500 and 1,000 different bacteria in your gut, And we have two different words now. We have commensal and we have symbiotic, right? Commensal bacteria could just be there. They don't necessarily elicit a beneficial effect, but not negative effect either. But a symbiont, by essence, elicits a beneficial effect on the host, and the host benefits the microbe. So that's a symbiotic relationship. So what we can do is discern the commensals from the symbionts using these germ-free mice by taking a germ-free mice and just colonizing with one bacteria at a time. And then you can look at the effect of that colonization of that one bacteria. It's called a monocolonization. And what you're looking for is the development of gut tissue. You're looking at the development of the immune system, the education of the immune system and how the immune system develops, and also how these mice digest food as well. Because just like I said, when you eat your plants and everything like that, that serves as a substrate to your gut bacteria, and indeed there are reactions in the gut bacteria that go on to release essential chemicals like vitamin D and things like that for our bodies. So there's the immune side, the tissue development side, and the metabolism side that we look at, and many other things like that we could go on, like bone development and behavior and liver cell development and all these things. So that's how we can discern which microbes in your gut can other symbionts that elicit the beneficial effects, and there might be others, commensals, that are just there for the ride.
Yeah, that's interesting.
And that stemming from that, finding out which ones are the symbiotic is the next step towards probiotic bacteria, right? So if we want to talk about have you discussed probiotic bacteria on this podcast often?
We've talked a lot about fermented foods.
Okay.
And a lot about fermented foods as potential sources of probiotics in the gut. I think many of the audience are familiar with recommendations to, eat live active yogurt cultures, when taking antibiotics and things like that. But before we jump to that, I just want to reiterate how amazing of a tool this is, that from this, incredible complexity of the gut microbiome, which has, thousands of microbes, many of which we can't even culture in the laboratory setting still. And the ability to go through and one by one look at these and see, what effects they have on, as you said, the education of the immune system. I think this is an important part, is that, our bodies learn, our immune systems develop over time and exposures, and a lot of it's through these interactions with these gut microbiota. Without those, we'd be in trouble, right? I mean, if we didn't have those interactions.
And absolutely in early life. So, you see a baby crawling on all fours along the ground, okay? And what is the natural instinct of the baby after that, or the infant, is to put their hand in their mouth, right?
The colonizing
all thoughts throughout where it's going to live, and then puts its hand in its mouth. So within, and this is, an ancient trait that babies have, and you see mothers saying, "Don't put your hand in your mouth," or something like that. But actually what probably is happening at that time is that we are sampling the bacteria in our environment. Putting a hand in your mouth, you're getting colonized with that one bacteria. Your immune cells in your gut and beyond are recognizing that bacteria, establish a tolerance to that bacteria, understand that bacteria is not pathogenic, it's not a foe, and therefore there are things in the immune system that allows us to live side by side with our bacteria in our environment like that. So there are little nuggets like that, like, the instinctive nature of a baby to put its hand in its mouth, right? What it's really doing is sampling the environment and, educating its body about the bacteria in the environment, educating its immune system to tolerate the bacteria in its environment.
Yeah, to recognize friend from foe, in other words, of which microbes are okay and which aren't. Well, getting to this point of, I guess, healthy microbes, let's talk a bit about probiotics. I mean, from the dietary supplement industry, probiotics are, make up a large portion of the economic share. Lots of people take these probiotic pills. A lot of people also eat fermented foods, whether it's in the form of fermented dairy or lacto-fermented vegetables, pickles, things like that. So, what do we know about these probiotic organisms, and are they really beneficial to our gut? What does the science say so far?
Bottom line, yes, they're beneficial to our gut if we choose the right ones. Okay, so
Mm.
that's the thing. So if a lot of the listeners of the podcast are involved with fermented food, then that really was the basis of the understanding of to modern day probiotics. So so you have the realization or the observation that populations, I think it was in Mediterranean populations, that ate fermented food, especially at an older age, they lived longer. They had a better health span, if you like. They were healthier at an older age. So that was some of the initial observations by Eli Mechnikov way in sort of back in the first decade of the 20th century, 1910 or so. Then, realizing that there are microbes that elicit a beneficial effect. And then I think Fuller back in the 80s described microbes as live microbial feed supplements. I think that was the next sort of definition of probiotics. And I think the latest definition for a probiotic is a live microorganism which, when administered in sufficient amounts, can confer beneficial effects on the host. So yes, my probiotic is a real thing. Now, identifying which ones are the probiotics, that's the challenge, okay? Mm. And that's a main focus of my lab. We have high throughput systems to test many different types of or candidate probiotics and see if they have positive effects on the gut, for example, and beyond on the liver as well. So we have identified. So what is interesting is that you can have two bacteria from the same taxonomic group, like two lactobacillus species, and one is probiotic and the other is not, or elicits a beneficial effect. So the beneficial effects of probiotics is strain specific. Some of those probiotics have something in them that it might be a bioactive molecule or something like that. And that bioactive molecule is absorbed and penetrates throughout organs and systems in our bodies and elicits the beneficial effect. So that's the next frontier in probiotics, and that's the addressing what is lacking at the moment in probiotic research is the actual mechanistic explanation of why probiotics are good for you. That is, what is released by the probiotic? How does our body recognize that factor that's released by the probiotic, either with receptors or anything like that? And then there are cell signaling pathways within our body, with cells in our body that are activated by this molecule from the probiotic, and somehow it elicits a positive effect. Wow. I'll give you one example, one cool example, and it pertains to your sort of fermentation group. So what we found is if you take a probiotic, a lactobacillus, those are lactic acid bacteria, and those are used in your food preservation, right? So we fed these lactic acid bacteria to mice, And what we saw that in the gut and in the bloodstream as well of mice, that the levels of butyrate, this short chain fatty acid, increased. And this butyrate is a fantastic compound. It elicits all these super beneficial effects on the host. Now, what we know is that the lactic acid bacteria can't directly make butyrate. So what was happening is that the lactic acid, you take the lactic acid bacteria, it goes into your gut, that makes lactate, and then it's other bacteria in your gut that converts the lactate to the butyrate, and then that butyrate is taken up by the host, and it affects the immune system, is anti-inflammatory, and it's the main food source for our gut cells, our gut colonocytes as well. So you have this, so you're giving the lactobacillus, the lactic acid bacteria, and what you're doing is making a food source to feed other bacteria in your gut, and then it's them that actually bioconverted the lactate to butyrate, and that was absorbed. So that's a mild degree of complexity, but it gives you an example of the type of thing that goes on within our gut as a result of eating beneficial bacteria.
That's incredible, Reynold, because on one hand, I expect nature to work like this, where it's not just one thing that's doing X, Y, and Z, but it's one thing doing X, and then another species comes and helps out with the next part. We see this a lot in plant chemistry, where you have these concepts of synergy of many compounds coming together to yield your desired, outcome. But this is even more complex because of multi-species involvement, both in the microbial level and the host level.
Absolutely
amazing.
And if you eat Brussels sprouts or kale or something like that, they're super rich in all the chemicals, and so your gut bacteria has this. It's a fantastic repository of metabolism. One step back, when you compare the number of bacteria in your gut and the number of cells in our body, it's a 1-to-1 ratio. But when you compare the actual genetic information, we have a certain amount of genetic information inside our cells. But if you take all the microbiome together, that genetic information that is in the microbiome is just like several magnitudes more than in our cells. And it's got this fantastic capacity, this metabolic capacity, to take the substrates that we give it when we eat your plant material. Mm-hmm. Your gut microbes will then bioconvert your plant material and perhaps make all these crazy exotic compounds that we absorb in our body. And we know that these compounds can permeate every organ, goes to the liver primarily first. It gets shunted to the liver along the portal vein, then goes to the heart, to the brain, and all over the body. So the potential mechanisms involved with, what is it? It's the food, microbe, and liver sort of axis, if you like, of food, microbe, and then bone axis, or food, microbe, brain axis, right? You could have chemicals produced by your gut that influence your mood, for example, or sort of gives you anxiety or calmness or everything like that. We know that chemicals sort of can do that. We don't know how many of them would be affected by plant, microbe, and then brain diet or axis, right? So that's a huge sort of potential avenue for research.
Oh, it's a tremendous avenue. We're talking about, when we go back to kind of the Hippocratic corpus and thinking about food as medicine, this is, on the very deep level, food as medicine, not only for physical health, but also mental health, when you're thinking about how, the mood modifying capacities of compounds produced by these microbes. It's still an area that needs a lot of research, right? Yeah.
We're only at the tip of the iceberg, because the challenge is identifying these small molecules. So, going back to the germ-free mice. So a germ-free mouse has 0 bacteria, but then what we call is a conventional mouse, a mouse that's replete with 1,000 different bacteria. So we did the metabolomics analysis of a germ-free mouse and a conventional mouse and found that the mouse with the livers, okay, of these two, the liver of a germ-free mouse and the liver of a conventional mouse that has bacteria, you will find that about 15 to 20% of the metabolites inside the liver of the conventional mouse is absent in the germ-free mouse. So about maybe 15%, 20% of the metabolites inside your body are derived from the microbes, are derived from biochemical activity of the microbes that aid in your digestion and in the metabolism like that.
Wow.
So, and it's actually, you know, you do mass spec and things like that. You know, when you think about 15% of all the metabolites in our body, that's thousands of metabolites, right?
Yeah.
You've got to choose one that to study and choose one that you're likely to show a beneficial effect or an impact on host physiology. So the limiting of the challenge in the field, it's that a daunting endeavor to do this kind of science because you have so many different metabolites produced by the microbiome. And which one do you choose? And you invest a lot of time in going after it. And if nothing comes of it, then that's a lot of wasted time, right? So that's the challenge at the moment in the field.
Yeah, no, the chemical complexity there is just, I mean, almost unfathomable, to try and chase this down on a, one compound at a time kind of endeavor.
Right.
Well, one thing you mentioned too, I wanted to make sure we talked about this. So we're talking about, monocolonization with single species. But you've also done some really interesting work with basically a humanized mouse by taking the human gut microbiome. So how does that work? How do we replicate as close as we can what a healthy human gut microflora might look like in one of these mice?
So absolutely. So when you ask healthy, that's different in each case, but the sort of rather than healthy, is what's been done in the science at the moment is a disease-associated microbiome.
Okay.
So there's a lot of, perhaps over the last 15 years, there's a lot of science where people have taken a cohort of diseased patients and then a healthy cohort, like 50 and 50. They've done the microbiome diversity of the diseased patients and then the microbiome of the healthy people. And then they try and see if they group together or anything like that. And then they can come to the conclusion that it's this microbiome diversity that causes the disease. Well, if you just take one snapshot of one disease patient, you don't know, you can't establish causality. You don't know whether that microbiome diversity is as a result of the disease, right? Especially in something like Crohn's disease or IBD, your microbiome is going to change because of the, crazy inflammation you've got going on in your gut. So to establish causality of a microbiome in human that people think is associated with a disease or causal of a disease, one way you can to do it would be to take that microbiome, transplant it to another animal, and see if that animal manifests that disease. But like I said, you can't just transplant it to something that's already got a microbiome, because those microbiomes that's established in your gut will fight off the what's being transplanted in. So that's why you need a germ-free mouse to do this. So you take, say, a human, your hypothesis is that this microbiome diversity causes obesity, for example, or something like that. So you collect that microbiome from the human patient, and then you go through some techniques and give it to a mouse. So give the inoculum to a mouse, and then you have to store that mouse in a hermetically sealed cage after that, such that no other microbes come in and disrupts that. So the mouse gets colonized with this human microbiome, And then in our experience, about 90% of that microbiome sort of grafts into the mouse, and the mice are very happy once they've got a human microbiome. They of course, they're on a mouse chow diet afterwards. So the experiment would be to keep feeding the human diet to the mouse, but we can't do that. We give them the mouse chow diet, and there is a little bit change in the microbiome. But then you start on the experiment, whether that microbiome alone would cause obesity in the mouse, or whether it's a combination of that microbiome and a high-fat diet or a westernized diet or something like that is the causality. So you can that's the approach of, if somebody wants to make the claim that this microbiome diversity in humans causes this phenotype in humans, well, that's one approach to prove causality, is take the microbiome out of the human, put it in a germ-free mouse, colonize, and then see if the phenotype is recapitulated in that organism. And of course, with the obvious caveats of, yes, it's a mouse and it's on a chow diet and things like that.
Yeah, I seem to recall a few years ago that experiment had been published, right?
Yeah,
and that the mice did become obese.
Yes.
I don't recall if it was under regular chow or high fat chow, but that the human microbiome was very important in that outcome.
Precisely, yes. I think with a regular chow, it was just a minor increase, but it was exaggerated manyfold with a Western diet. So yes, those studies have been done.
So now the real question is, when are we going to get the skinny microbiome transplant? I'd like one of those. With your high-fat diet.
So this brings us back around to probiotics, okay?
Yeah.
So if you've got a microbiome diversity, it's got 500 to 1,000 different gut microbes in there. If you do identify a certain 50 microbes or whatnot that causes obesity or makes obesity worse, what do you do afterwards? You can't really go in and remove them, right? Because how would you do that?
They're entrenched. Yeah.
You basically we talked about antibiotics. You can't have antibiotics that are specific for those microbes. Perhaps you could, in the future, perhaps 10, 20 years ahead, is going to be phage therapy that might get rid of certain taxa. That's a more specific way of getting rid of certain microbes out of the community. But instead of trying to manipulate your extant microbiome, the microbiome that's there already, what you do is use the bacteria therapy, the probiotic bacteria, and then you identify a probiotic bacteria that is effective in against the bad effects of a Western diet. And we had a paper last year that identified one certain bacterium. It's a Lactococcus lactis, and we don't know the mechanisms of how it does this yet, but when we fed mice a Western style diet that was high in sugar and high in fat, so that's important, because I think some studies just put a high fat diet and they forget about the sugar.
Very important.
Precisely. So if you give mice these high sugar and high fat diets, they get big globules of fat in the liver. But we fed this probiotic as well over a period of 10 weeks, and the mice that got this specific probiotic and not other probiotics that we also included in the experiment, those mice, they gained significantly less weight, and they had none of these fat droplets in the liver. Wow. So we published that last year in Gastroenterology, one of the AGA, the American Gastroenterology Association's flagship journal. So if you get the right one, Cassie, the right gut microbe or the right probiotic, it might elicit the positive
effect. So when it comes down to strain types, is this strain commercially available? I mean, is this something that the general public can access?
It's not available yet. So what we did, it's a type strain. So what we did, so there is a repository of bacterial strains that have been characterized. It's called the ATCC, the American Type Culture Collection. And we bought, dozens of strains from there, candidate probiotics. We put them through our discovery platform to look for their probiotic influences and identified this one that was elicited beneficial effects on the gut. It protects against inflammation in the gut and also protects against the accumulation of fat in the liver as a result of a high-fat diet, or a Western diet, I should say. Wow. So we did a screen first to identify this one. And then we used this one in mouse experiments because that's the one thing you could never do a screen. I would never want to do a screen in mice, right? That's a bad idea. So once you do one with that works in another model organism, then you can use that one and perhaps do directed experiments in mammals. And
you did this with flies often, right? With the fruit fly. Yeah.
Precisely. That was our screening platform. So there is certain conservation in the beneficial microbes that exist in Drosophila, in fruit flies, and in mice and humans. So just the same that we can make germ-free mice, we can also make germ-free flies. Okay.
That's so cool.
So we make germ-free Drosophila, germ-free fruit flies, and then we monocolonize those Drosophila one by one. And of course, experimental wise, you can get great power in your experiments, lots of replicas, right? Because they breed like flies, right? You can have, so we would take a couple hundred flies, monocolonize them with a candidate probiotic, and then we would give these flies an insult that would cause an injury in the gut. And then we would look at the gut and assess how the flies would survive in response to this injury. And a lot of bacteria, a lot of these candidate probiotics did nothing, but this one probiotic and a couple of others, they elicit, the flies survived in response to this insult that we used. And then we took just those ones that were positive in the flies. We looked at them in cell culture, in mammalian cell culture to start off with. And once we got some positive data from that, we thought, okay, this is a real positive candidate for a new probiotic. And that's when we, had the justification to go over into the mouse experiment.
Mouse model. So this is really a systematic, kind of very intricate, detailed process that I guess, I wonder what your thoughts are on next steps. How does this process work when you identify potentially health benefiting probiotics? Is there a clear path to market? Because I can just imagine what all the listeners are thinking right now. They're shouting at me like, what are the best ones to take? What do we do? Like, how do you sort through all these products are available, or maybe even not go to products, but are there certain foods, fermented foods, that we should be adding in greater level to our diets?
And that's where we're at the moment, Cassie. So I'm yelling the same questions as well.
Yeah.
Something about it. So absolutely. So it's the regulation, right? The FDA regulation for the probiotics at the moment. If your listeners can read up on it. So if you Google FDA regulation probiotics, there's no, as I understand it, no probiotic bacteria that's had that's been cleared to as a therapy for a disease, right? So they are treated as supplements, just in the same class as if you go to your local store and buy some vitamin D or some a supplement in that respect. So yes, the next step would be to actually do a clinical trial and have a cohort of disease patients and a cohort of healthy patient, give the probiotic and see that there's a beneficial effect of the probiotic. And then, of course, you have to make sure that in the healthy cohort that it does no harm, right? So
Yeah.
well, it's just like, the coronavirus vaccine. That's what we've heard about recently. You have the clinical trials that needed to be done for that. But those clinical trials have to be financed, right?
Yeah. This is
Cost.
always the problem with science. These kind of studies cost a lot of money to run in humans.
And going back, because it's topical at the moment, you could do the coronavirus vaccine because the money was clinical trials, because the money was there, the impetus was there. There were other vaccines against other viruses that those companies developed, but they didn't. The clinical trials hadn't been done yet because the money was not behind them to do those clinical trials. And we're talking about tens of millions of dollars, for something like to get a product like this out to market. So that's why the probiotics remain as like a functional food, like a supplement in that class, because like you asked, the big step would be to do a clinical trial costing millions of dollars, and you need somebody to take the risk of putting their money down to fund it and see if there's you get an outcome. So before doing those clinical trials, what those companies need is what they call preclinical evidence that something works, like a probiotic works on a disease model. And what we're trying to gain now, our preclinical model is a mouse model. Okay, so you do these investigations on fatty liver disease in mice or IBD in mice and things like that, and you show that your probiotic would be protective in that realm. But you need a company that's brave enough to then go from just to realize, the difference between mice and humans. And mice, they live in the cage. Their microbiome is homogenized. The 5 mice that you would have in a cage, their microbiome is all the same. If you have multiple cages, you can have differences in the microbiome of mice between cages, but at least they're all on the same food. Okay, I told you all that because if you want to translate that to humans, the heterogeneity is immense in the microbiome diversity. Everybody has their own microbiome diversity. And if you think that this extra bacteria that you're going to feed, it might work in some patients that has a certain microbiome diversity and not in others. There's all these variables, all this heterogeneity that's inherent in our in society. And I'm sure you've talked about it before, is whether they eat, the diet is a factor, whether they've taken antibiotics is a factor, the cleanliness of the house, whether they have the dog, and whether they have kids and everything like that. So that also would be a challenge to the next step in taking probiotics from supplements, from functional foods to actual, approved, FDA approved
therapies.
Yeah.
And medicines. That's amazing. Well, I mean, I think you've given us a lot of things to think about, starting with the evolutionary perspectives of, multicellular organisms being so intricately intertwined with microbes. I mean, and that goes, as you said, going back billions of years. And, really great perspective on the complexity of these questions. Even in a controlled laboratory setting, it can get really complex. But as you said, in humans, we have so many different variables, different microbiomes, different foods people are eating, exposures. It gets really, really complex. But at the same time, I'm just so excited about this field of science. And I think that, when it comes to the future of health, I think that understanding the role of the gut microbiome is going to be fundamental, for the future of human health.
Absolutely. And like I said, it's these little molecules that are produced by the gut microbiome, these thousands of different molecules. And like I said, we're only at the tip of the iceberg of identifying how these bioactive small metabolites produced by the microbiome enter throughout our bodies, including gestating babies in utero. So things like that. So just to mention, we just got a paper accepted, Cassie. So basically what we did was, I mentioned this butyrate compound before. So this is a compound that's made by bacteria. So we took some pregnant mice, we supplemented them with butyrate and with this microbe, our new probiotic. And just during pregnancy, we let the babies be born, and we let them age those offspring until they were 6 weeks old, and those offspring were protected against gut inflammation. So so we're just that was accepted in pediatric research recently. So so not only do these gut microbe metabolites affect us, but they can permeate, and that's an example of how a gut metabolites can permeate throughout our body for certain, but it crosses the placental barrier as well into a developing fetus, if you like, and causes beneficial effects on that. So that really, I think, is the next 20 years in microbiome research, is identifying the small molecules produced by the microbiome and how they affect our health and disease.
Amazing. Yeah. Not only what your health, but for gestating mothers, like what's going on with the baby. Wild.
And you pass it on to the next generation, via the gut microbe, that sort of, that's the idea.
Well, as we wrap up, Rheinallt, I'll ask one last question. This is one I ask of every guest, and I was wondering if you can share with us one of your favorite recipes. I know you're from Wales. Maybe you have a traditional Welsh dish, or maybe a fermented food. I don't know. What's your favorite food to share with the audience?
Well, of course, there's no other dish but Welsh lamb, right? And I think you might taste some pretty soon if you're taking a vacation in Wales. So when you go over there, do have some Welsh lamb. So, of course, I spent my formative years in Wales. I was raised on a sheep farm in Wales, and I see these lambs eating grass, and they're all natural and everything like that. And my favorite cut of the lamb is the shoulder. So lamb shoulder, and in Wales we used to roast the whole ram shoulder. But when I came here to the States, you guys like grilling, of course. Grilling's not such a big thing in the UK, or perhaps it is now, but grilling is obviously a big thing over here. So one of the dishes that I take is have lamb shoulder steaks. Okay.
Nice.
Then I'd do a simple marinade in just your regular lemon juice, olive oil, salt and pepper, and rosemary, and some garlic as well. And then you leave that for about 2 hours or so, and then bring them out, pat them dry, and then have a searing hot grill and throw them on the grill. And it's better if your chops has some bones still in it, such that you know that they're cooked when the meat falls off the bone. That's what you look for. And then, so me and my wife tasted some of this on Saturday night, and she was like, wow, it's like, how did you cook this? Because, guys in the States over here don't eat so much lamb. Yeah. So, but when it's, hoping that I took the best of my knowledge in Wales about the best cut of lamb, and then I cooked it more American style, on the grill, and it was really tasty. And of course, with some homemade mint sauce, right? We have a big mint plant outside. Take some mint, and then you grind it in some salt, add some red wine vinegar, some good quality olive oil, and just make a paste of it. And that's your own mint sauce. Oh my gosh!
My
So,
mouth is watering. That sounds so good. That sounds great.
so
Well,
I hope you get a chance to sample some of that on your vacation.
yeah, I hope so too. I hope so too. And for the audience, I'll definitely be posting some photos come the end of October, early November. I'll be traveling into Wales and including Snowdonia National Park, which I'm really excited about seeing. Really beautiful. Well, thank you so much, Reinaldt, for coming on the show. This has been just incredibly insightful to understand the complexities of the gut microbiome. And I look forward to reading more about your research in this area.
Thank you for having me, Cassie.
Right. You've been listening to Foodie Pharmacology, the science podcast for the food curious, recorded on Skype. You can find this and all of our other episodes on Apple Podcasts. You can also check out the video version of this episode at the Teach Ethnobotany YouTube channel. Thanks so much to our producers, to Rob Cohen and Christine Roth, and thank you to you, our audience, for tuning in each week. Stay healthy out there, and I'll see you next time.